Welcome to the deep dive. Today we are embarking on, an absolutely incredible journey, not just into the building blocks of life, but into the very engine that powers them.
Mhm.
We're stepping into the vibrant, intricate world of biochemistry, that hidden symphony of chemical reactions that defines what it means to be alive.
It really is a symphony.
Yeah. Our mission with this deep dive is to sort of distill the essence of these chemical processes, the ones that underpin all living organisms, providing you with, hopefully, a genuine shortcut to being truly well-informed about these invisible forces shaping your world.
Definitely.
We've got a pretty comprehensive exploration ahead of us, so okay, let's unpack this.
And what's truly fascinating here, I think, is that by the end of this, you won't just have like a list of facts.
Right.
You'll gain a clear, structured understanding of how chemistry actually animates life. Connecting concepts that might seem totally separate otherwise.
Those aha moments.
Exactly. We're aiming for those aha moments that come from really grasping the fundamental mechanisms. And that's just profoundly important for comprehending everything from our personal health and nutrition to the big challenges in our environment and even like the future of technology.
Absolutely. So much relies on this. Okay, let's jump right in. Part 1, the foundations. Let's kick things off with the origins of this incredibly important field. When did biochemistry really, carve out its own identity as a distinct science? What was that pivotal moment?
Well, the term biochemistry itself is surprisingly modern, actually. It was first introduced by German chemist Carl Neuberg back in 1903.
1903, wow.
Yeah, think about that. In the grand scheme of science, this field gets into the very core of life. It's barely over a century old as like a formally named subject. Before that, all its insights were kind of scattered across other bits of chemistry.
So a 20th century term for a science that feels almost as ancient as life itself. And how did they formally define it back then to sort of separate it from just general chemistry?
It was defined quite precisely, actually, as a science treated with the chemical nature and their behavior of the living matter.
Okay, sounds a bit academic.
It does, yeah, but let's break that down. It's not just about what chemicals are in living things, but really how they behave,
Ah, the behavior.
Right. So the studies associated with it, they specifically look into the chemical elements you find in biological systems, their transformations, like how they change and react within those systems, and crucially, the energy changes that are intrinsically linked to those transformations. It's all about the dynamic, constantly shifting chemical landscape inside a cell.
That's a really precise definition. And where are these incredibly detailed studies actually carried out? Are we talking specific biological context, or is it like broader?
Oh, it's incredibly broad. These studies have been meticulously done in both animal and plant tissues.
Both.
Yeah, this universality across all living things is exactly what makes biochemistry so foundational. It doesn't matter if you're looking at a tiny bacterium, a huge redwood tree, or, the basic biochemical principles are always at play.
That's why it's so foundational, right? It applies to everything alive. Okay, so how does this connect to our own bodies, our health, in a really tangible, immediate way? Because I think that's what a lot of people connect with biochemistry.
That's a great point, and it really highlights one of biochemistry's most direct impacts. It's so intimately tied up with human health that it's often considered basically a branch of human physiology.
Right.
Think about your last doctor's visit if they took blood or asked for a urine sample. They were likely tapping right into biochemical analysis. When we perform chemical analyses of bodily fluids, blood, urine, other natural secretions, we're essentially watching biochemical processes in real time. And this analysis can directly help diagnose all sorts of diseases, offering this kind of profound chemical window into our health.
Like detecting high sugar for diabetes.
Exactly. Detecting abnormal glucose levels points straight to diabetes, or specific enzyme levels might indicate liver damage. It's just invaluable for understanding and treating illness.
It's incredible how a science formally named in the early 1900s has such immediate practical uses in a doctor's office today. It literally gives us insights into our own internal chemistry. But how did the field itself come about? You mentioned its roots are sort of distinct, evolving from other areas.
Yeah, it didn't just pop into existence fully formed. Biochemistry actually started out as a kind of specialized branch of organic chemistry, the chemistry of carbon compounds.
Okay.
But what really gave it its power, its interdisciplinary nature, was later on when it integrated ideas and methods from physical chemistry.
Ah, the physics side.
Right. This combination allowed biochemists not just to identify molecules, but to understand the forces and the energies that govern how they interact. That made it a truly comprehensive science, drawing from multiple foundational areas.
That explains why it feels so comprehensive. It's like a scientific melting pot. And I know there were some early pioneers who really set the stage even before the name biochemistry existed. Who should we know about?
Yeah, two figures really stand out, each giving us a crucial piece of the puzzle. First, there's Carl Wilhelm Scheele. He was an early 18th century Swedish pharmacist.
A pharmacist. Interesting.
Yeah. Scheele was incredibly meticulous, a real observer and experimentalist. He painstakingly figured out the chemical composition of all sorts of drugs, plants, animal materials. His work was groundbreakingly specific for the time. He was the one who identified things like lactic acid from milk, malic acid from apples, citric acid from lemons, even uric acid from urine.
Oh.
His tireless efforts in isolating and characterizing these biological substances were so significant, he's rightly considered the founder of descriptive biochemistry. He basically gave us the initial inventory of life's chemical parts.
Okay, here's where it gets really interesting, that distinction. Descriptive, which Scheele pioneered, focusing on what chemicals are there, and then there's dynamic. So Scheele was about what makes up life. Who was the other key pioneer, and what did they bring to move beyond just like an inventory?
That would be Antoine Lavoisier, a brilliant French chemist from the late 18th century. Lavoisier fundamentally changed how we understand chemical reactions altogether.
Yeah.
He famously studied the composition of air, developed the crucial concept of oxidation, combining with oxygen.
Right, oxidation.
But for biochemistry, his most profound contribution was explaining the nature of animal respiration. He showed that breathing is basically a slow combustion process. It consumes oxygen, produces carbon dioxide, much like a burning candle.
Revolutionary.
Totally revolutionary. Because it showed that living processes followed the same fundamental chemical laws as non-living ones. For these insights, Lavoisier is known as the founder of dynamic biochemistry, and he's also widely seen as the founder of modern biochemistry itself. He moved beyond just identifying compounds to explaining how they change, interact, and generate energy within living systems.
So as Scheele identified the players, Lavoisier started figuring out the rules of the game, how the game generates energy. That's a huge shift. And speaking of the game, at its heart, what is the very nature of biochemistry, this chemistry of life? What defining characteristics of living things does it fundamentally explain?
Well, at its core, biochemistry is fundamentally the chemistry of living things. When we look at living organisms, biochemistry helps us understand that amazing array of characteristics that define them, like their fundamental ability to move, to grow, to respire, which Lavoisier started to figure out to respond to stimuli from their environment, and crucially, their capacity to reproduce. At a molecular level, living things are incredibly complex and structured. They contain this immense number of diverse organic molecules.
Yeah.
Each one meticulously designed with a specific job. And of course, a hallmark of life is the ability to acquire, transform, and utilize energy, whether that's from nutrients they eat or directly from sunlight.
Right.
But among all these incredible traits, the capacity for self-replication, the ability to make copies of themselves, is often presented as the most important attribute. It's what ensures life continues generation after generation.
How does understanding these incredibly intricate chemical underpinnings change our perspective on life itself? It really makes you appreciate the elaborate molecular machinery, doesn't it?
It absolutely does. It shifts our understanding from just looking at living things from the outside to this deep appreciation for the elegant molecular ballet happening constantly inside every single cell. It reveals that life isn't just some mysterious vital force. It's a meticulously orchestrated, highly regulated series of chemical reactions and interactions. It lets us see the fundamental similarities across all living things. At the molecular level, the same molecules, the same reactions, the same energy currency, like ATP, even while they express themselves in this astounding diversity of forms and functions. This perspective opens up whole new ways to understand and maybe even manipulate life processes.
Given that it sounds like biochemistry is one of the most important sciences today, even though, as you said, it's relatively new as a named field, it's essentially the bridge between the non-living chemical world and the living biological world.
It is indeed. Biochemistry is, comparatively speaking, a new branch of science, with much of the foundational work and organized understanding really developing extensively just during the last century or so. Its core importance is precisely its ability to bridge that gap, to fill the void between the highly complex functions we see in living cell, like thinking or muscle movement, and the different, sometimes seemingly simple properties of its chemical elements.
Right.
So a biochemist then is like an examiner, using this diverse toolkit, chemical, physical, biological techniques to research the chemical nature, the constituents, the behavior of living stuff. They undertake this research with, ideally, sincerity, patience, and honesty. They're basically the detectives of life's molecular mysteries.
So what does this all mean for us then? Where do we actually see biochemistry at work in the real world, beyond the lab or the doctor's office? How does it impact our daily lives?
Oh, its crucial role spreads across so many fields, truly touching every aspect of our lives, often without us even noticing. In medicine, for example, the impact is just profound.
Yeah, we mentioned diagnosis.
Right? Disease diagnosis through blood tests, urine tests, even complex genetic analysis. They all fundamentally rely on biochemistry to spot abnormalities in proteins, enzymes, metabolites. Think about a genetic test for, say, a predisposition to certain cancers. That's biochemistry tracing specific genetic markers. Drug development is maybe one of the most direct applications. Pharmaceuticals are designed based on our biochemical understanding of how diseases work, often targeting specific enzymes or receptors in the body.
Of finding the target.
Exactly. And biochemical studies are absolutely vital for identifying and treating genetic disorders like cystic fibrosis or sickle cell anemia, which are at their core molecular diseases, biochemical errors.
So from finding out what's wrong to finding new ways to fix it, biochemistry is right there.
Exactly. And its reach goes way beyond human health. In agriculture, biochemistry underpins tons of modern advancements. Think about GMOs, genetically modified organisms.
Right.
Biotechnology, which is built on biochemical principles, helps produce crops resistant to pests and diseases, like bright corn that makes its own insecticide. Understanding plant biochemistry also helps develop more efficient fertilizers and targeted pesticides, trying to minimize environmental impact. And biochemical analysis of soil is crucial for keeping soil healthy, for sustainable farming, making sure nutrients are there for future harvests.
From the clinic to the farm, that's a huge span.
It is. Then there's nutrition and food science, which directly affects our daily well-being. Biochemistry explains in detail how our bodies process carbs, proteins, fats, vitamins, minerals, basically, how we get energy and building blocks from food. Biochemical techniques are also fundamental for food storage and preservation, extending shelf life, keeping food quality up. And a deep understanding of biochemical pathways helps design specific diets to prevent and manage chronic diseases like diabetes, obesity, making dietary advice much more precise.
That's literally on our dinner plates every day, informing what we eat.
And beyond that, in the exploding field of biotechnology, enzyme technology is huge. Enzymes are harnessed for countless industrial uses, brewing beer, making cheese, producing pharmaceuticals.
Right.
Genetic engineering, manipulating DNA, relies entirely on biochemistry. This gave us breakthroughs like gene therapy and mass production of synthetic human insulin for diabetics. Biochemical techniques also have broad industrial uses in biofuels, biodegradable plastics, even environmental management through bioremediation.
Wow, that's a massive span of influence, and it even extends to environmental challenges.
Yes, profoundly so. In environmental applications, biochemistry is key to understanding and tackling complex problems. Consider biodegradation. Microorganisms use specific biochemical reactions to break down pollutants in soil and water.
Cleaning up messes.
Pretty much. Biochemical processes are critical in wastewater treatment and pollution control, converting harmful stuff into inert substances. And even in the critical area of climate change mitigation, developing biofuels and carbon capture technologies relies heavily on fundamental biochemical principles to convert and store carbon.
So, from the incredibly precise molecular interactions inside one cell to global challenges like climate change, biochemistry truly permeates every facet of our world. It's absolutely pervasive. Okay, now let's transition from the broad field itself to arguably the most fundamental substance in biology, water. It might seem like just, a backdrop for life, but why is water everything in the context of living systems? What makes it so indispensable?
Yeah, water isn't just everywhere. It's the main solvent for all forms of living things. It's often described as, like, the vital essence, a miracle of nature, the great supporter of life, because its unique properties allow life to exist and thrive. At its core, it's the solvent where virtually all the chemical reactions of biological processes happen. Think of it as the universal stage where the drama of life plays out.
Right.
The very existence and function of cell structures depend entirely on water's unique physical and chemical properties. In fact, it's believed water was the first medium where cells arose, and it's remained the solvent in which most biochemical changes evolved over billions of years.
Well,
But its role goes beyond just being a reaction medium. It's also crucial for carrying stuff through cell membranes, maintaining the stable internal temperature life needs, and efficiently getting rid of waste products from organisms.
So it's not just a backdrop, it's an active, indispensable participant in every single process. It's remarkable how fundamental it is.
Exactly. And its unique physical properties really set it apart from other common solvents. For instance, compared to similar compounds, H2O has an unusually high boiling point, a high melting point, a higher heat of vaporization, and higher surface tension. These aren't just, trivial differences. They are directly responsible for water's life-supporting roles.
Fascinating. And what about vaporization? How does that specifically relate to life?
Right. The vaporization of a liquid, its change to a gas, is called evaporation. For ice, the direct jump to gas is sublimation. Both processes, evaporation and sublimation, increase rapidly as temperature goes up. Evaporation happens when water is heated above 100 degrees C at sea level. Critically for life, the reverse process, condensation, happens when heat is lost. This cycle plays a vital role in evaporative cooling for organisms.
Like sweating.
Exactly, like sweating in humans. As water evaporates from our skin, it carries away a significant amount of heat, effectively cooling the body, preventing overheating, which is crucial for keeping our internal temperature stable so our enzymes can work properly.
And just how much of our bodies are actually water? We hear that percentage thrown around a lot.
Yeah, in humans, water makes up a massive chunk of us. About 60% of red blood cells are water, and an amazing 92% of blood plasma is water.
92%.
Yeah. Other tissues are typically around 75% water. It just highlights how pervasive and essential it is inside us. And just to give another perspective, if we look at seawater, it has a minimum salinity of about 24.7 parts per thousand. Interestingly, its density decreases at higher temps and increases at lower temps, down to about 4 degrees C. Then it does something unusual. Also, there's no single freezing point for seawater because of all the dissolved salts.
These unique properties aren't just curiosities. They literally dictate how life functions at a fundamental level. And it's all down to how the water molecule itself is put together, isn't it? That H2O structure.
Indeed. Let's dive into that molecular structure, H2O. Each hydrogen atom forms a covalent bond with the oxygen atom, meaning they share electrons.
Right, sharing.
But the oxygen atom also has 2 pairs of unshared electrons, often called lone pairs, which really influence its shape. These electron orbitals create a rough tetrahedron shape around the central oxygen. But the HOH bond angle isn't perfectly tetrahedral. It's actually 104.5 degrees, a bit less than the ideal 109.5 degrees.
And this slight difference is key, right? It creates something called polarity.
It absolutely is. The reason is that oxygen is way more electronegative than hydrogen.
Meaning
its nucleus pulls shared electrons much more strongly than hydrogen's does. This unequal sharing creates an uneven distribution of charge across the molecule.
Ah, okay.
The hydrogen atoms end up with a partial positive charge. We note that as all or plus because the electrons are pulled away from them. And the oxygen atom bears a partial negative charge because it's hogging the electrons. This results in the water molecule having 2 distinct electric dipoles, making it a highly polar molecule.
This subtle arrangement of partial charges has profound implications for how water interacts with everything else in a living system. It lets water dissolve so many different things.
Which leads us right to the idea of ionization in water.
Exactly. While water's amazing solvent properties are largely down to its uncharged H2O molecules, water molecules also have this minor but critically important tendency to undergo reversible ionization.
Meaning they split apart. Yeah, a tiny fraction of water molecules can spontaneously split to produce a hydrogen ion, H plus A, which is basically just a proton, and a hydroxide ion, OH. This reversible reaction is written as H2O, H plus OH.
And this reversible ionization is really important for cellular functions, correct? Because it allows for fine-tuning acidity and basicity.
It is absolutely essential. This self-ionization is the very basis of the pH scale. When weak acids or weak bases dissolve in water, they disperse by either donating H plus ions like acids do when they ionize, or by consuming H plus ions like bases do when they get protonated. These processes are always governed by equilibrium constants, which mathematically describe the relative concentrations of reactants and products when the system hits a stable balance. For a general reaction like A plus B, C plus D, the equilibrium constant reflects these concentrations. And technically, it's represented using molar activity, which accounts for the effective concentration rather than just molar concentration, the total amount. Seems like a small detail, but it's crucial for accurate biochemical math.
So how do we measure this incredibly important H plus concentration in, say, our cells to understand its acidity or alkalinity?
Right. In 1909, a Danish biochemist, Søren Sørensen, introduced this brilliant logarithmic scale for expressing H plus concentration. And that scale is universally known as the pH scale.
Logarithmic, meaning not linear.
Exactly. It's critical to understand it's logarithmic, not arithmetic. This means each full pH unit represents a tenfold change in H plus concentration.
Tenfold.
Tenfold. The P in pH stands for power specifically. The power of 10 in H is for the concentration of hydrogen ions. Sørensen formally defined pH as the negative logarithm of the hydrogen ion concentration.
So to put that into context, pH 7 is neutral, like pure water, but pH 6 is 10 times more acidic, and pH 5 is 100 times more acidic. It's a very powerful way to measure these tiny concentrations.
Precisely. The pH of aqueous fluids typically ranges from 0, extremely acidic, to 14, extremely alkaline. This scale is absolutely fundamental to understanding virtually every aspect of biochemistry. Enzyme activity, which often needs a very narrow pH range, maintaining stable cellular environments, even diagnosing disease states, even small shifts in pH in our bodies can have drastic, even life-threatening consequences. So pH regulation is a critical balancing act.
Okay, let's zoom out a bit now, moving from the incredible properties of water to its building blocks, atoms. These are the fundamental units of all matter, and their structure dictates everything we've just discussed, right? From water's polarity to the complex reactions of life.
Indeed. At their core, atoms are the very small particles that form the basis of all chemical reactions. At the chemical level, you can't generate them, destroy them, or subdivide them further. A key principle of atomic theory, foundational to all chemistry, including biochem, is that atoms of the same element have identical properties and equal weight, while atoms of different elements have different properties and distinct weights. This fundamental difference gives rise to all the chemical diversity we see.
And what's inside these atoms? What's their internal architecture that gives them their unique identities?
The modern picture comes largely from Ernest Rutherford's groundbreaking model from 1911.
Rutherford, okay.
Yeah, his model says an atom has a dense central nucleus, positively charged protons, neutral neutrons. And orbiting this nucleus are negatively charged electrons, moving in discrete energy levels called orbits or shells.
Shells.
Right. These electron shells are numbered 1, 2, 3, up to 7, getting further from the nucleus and having higher energy. They're also traditionally classified by letters, K, L, N, O, P, Q, with K being the innermost. It's the electrons in those outermost shells that mostly determine how an atom behaves chemically.
So this microscopic world, the arrangement of these tiny particles, dictates everything from how water acts as a solvent to how huge proteins fold into their specific shapes. It's truly the foundational architecture. And sometimes atoms lose electrons, like in ionization. How much energy does that take?
That brings us to ionization potential, or ionization energy. It's defined as the amount of energy needed to remove the most loosely held electron from a normal, uncharged atom in its gaseous state. It basically measures how tightly an atom holds on to its outermost electrons.
Okay.
Symbolically, we show it as A, the atom becoming A plus the positive ion plus an electron E, and EP represents the energy needed to pull off that one electron. This energy value is crucial for understanding how atoms form ions and react.
Now, how do these atoms actually stick together to form molecules like water, or the much bigger biomolecules like DNA and proteins? That's where chemical bonds come in, right? The glue of molecules.
Exactly. Chemical bonding theory, explaining how atoms join up, was first really defined comprehensively by the American chemist G.N. Lewis back in 1916. Lewis, yeah. His theory says bond formation basically involves either sharing or completely transferring a pair of electrons between two combining atoms.
Sharing or transferring.
Right. The underlying idea is simple. The electrons in the bond feel a strong pull from both nuclei, that positive negative attraction which holds the two atoms together stably. This attraction is what we call a chemical bond, usually involving one or more electron pairs. Fundamentally, a bond is just any force holding two atoms together, creating a more stable setup by redistributing their electrons.
So it's all about reaching a more stable state by arranging those electrons.
Exactly. And based on how those electrons are redistributed, there are three main types of chemical bonds vital in biochemistry.
Okay, let's start with the first one, the ionic bond. What's that about?
The first type is the ionic or electrovalent bond. This involves the complete transferring of one or more electrons from one atom to another.
Giving and taking.
Right. This creates oppositely charged ions that then attract each other strongly. It's like one atom gives an electron away, becomes positive, the other accepts it, becomes negative, and boom, they stick. Ionic bonding is very common in inorganic compounds. The classic example is sodium chloride, NaCl, table salt. Sodium atom donates an electron to chlorine, forming Na+ and Cl ions held together by that strong electrostatic pull.
So a simple transfer, yet incredibly powerful in making stable compounds. All about stability. What about the second type? This one seems even more central to life.
Yeah, the second and maybe the most pervasive bond in organic and biological chemistry is the covalent bond. It's also called a nonpolar bond if the electrons are shared equally. This bond involves sharing one or more pairs of electrons between the atoms.
Sharing, not transferring.
Right, they become partners in sharing. Compounds formed this way are called nonpolar or covalent compounds. This type of bond is everywhere in organic compounds and plays a critical foundational role in all biomolecules.
Like where specifically?
Okay, so in proteins, the backbone is formed by peptide bonds. Those are covalent amide bonds linking amino acids together, forming the basic structure of enzymes, structural proteins like collagen. In nucleic acids, DNA and RNA, it's phosphodiester bonds that covalently connect the nucleotides, creating those stable, long chains needed for genetic info. And lipids, ester bonds covalently link fatty acids to glycerol, forming triglycerides, our main stored energy. And covalent bonds are also super important in enzyme activity. The making and breaking of these bonds drives countless metabolic reactions.
This sharing mechanism is how life builds its most complex structures, from tiny enzymes to huge DNA molecules. It's the fundamental engineering principle of biology.
Okay, and what's the third type of bond? A bit more specialized, maybe.
The third type is the coordinate bond, also called a dative covalent bond. What's unique here is that one atom donates both electrons needed to form the stable interaction with another atom.
One provides both electrons. Exactly. It's still sharing, but the electrons come entirely from one partner. These bonds are absolutely essential in many biological processes, especially involving metal ions.
Metal ions, like iron.
Precisely. Like in metal ion complexes in enzymes. A prime example is hemoglobin in our red blood cells. An iron ion, Fe(O), forms coordinate bonds with nitrogen atoms in the heme group, which allows it to reversibly bind and carry oxygen.
Okay.
Many enzymes needing metal ions rely on coordinate bonds for their catalytic action. Carbonic anhydrase uses a zinc ion, ZOO. Cytochrome C oxidase uses iron and copper ions, Fe(O) and CuO, coordinated to shuttle electrons. Even the stability of DNA and RNA depends on ions like magnesium, MCO coordinating with their phosphate groups, helping with replication and transcription. Plus, many cofactors, helper molecules for enzymes, use them. Vitamin B12, cobalamin, uses coordinate bonds to a cobalt ion for metabolic reactions. And these bonds facilitate oxygen transport in some critters via hemocyanin, using CuRO, and are crucial for photosynthesis in chlorophyll, using mGRO to capture light. So, if we connect this to the bigger picture, these coordinate bonds are truly the silent architects behind so many of life's most critical functions. They allow precise binding, efficient electron transfer, structural integrity, enabling processes vital for every living cell.
That's incredible. So much of what happens breathing, digesting, depends on these tiny, precise interactions, these specific ways atoms are glued together. All right, let's transition now to the first major class of biomolecules we encounter, carbohydrates, often just called sugars, right? What exactly are they, and why are they so incredibly abundant?
Yeah, carbohydrates are indeed termed sugars, or saccharides for most organisms. The name comes from the Greek saccharon for sugar, and based on sheer mass, they are the single most abundant class of biomolecules in nature. Just tons of them out there.
Wow.
These molecules are primarily composed of monosaccharides, disaccharides, and polysaccharides, basically ranging from simple single units to really complex chains. They're widely distributed in both plant and animal tissues, though, interestingly, plants are much richer in carbs than animals. They form plant structural bits and energy stores.
And what's their core composition? Just CH and O, right? And what's their primary role, beyond just being sweet?
That's right. At their most basic, they're biomolecules made of carbon, hydrogen, and oxygen, often in a ratio that looks like hydrates of carbon, hence the name. Their primary, most well-known role is definitely serving as a major source of energy for living things, quick fuel for cells. But they're way more than just energy packets. They play huge roles in overall metabolism, participating in countless biochemical pathways. They're crucial for cellular communication, acting like ID tags on cell surfaces so cells can recognize each other. They also provide essential structural integrity, think plant cell walls, insect exoskeletons.
Right, structure too.
Yeah. Plus, they participate in critical processes like DNA and RNA formation. They form the sugar part of the backbone. And as mentioned, they contribute to immune function and cell signaling through specialized molecules called glycoproteins and glycolipids on cell surfaces. Overall, carbs are absolutely indispensable for sustaining life and maintaining that whole intricate balance of biological functions.
So not just quick energy, they're structural, communicative, foundational for genes. How are these multifaceted molecules, from simple sugars to complex starches, actually classified? How do we organize them?
They're generally classified into three main groups based on their complexity, how many sugar units they have. First up, monosaccharides. These are the simple sugar.
These sick units.
Exactly. The simplest form can't be broken down into smaller sugar units by hydrolysis. They typically have a free aldehyde CHO group or a ketone CO group, plus two or more hydroxyl groups. They're the building blocks. Monosaccharides can be further subdivided by how many carbons they have. Triose is 3, tetrose is 4, pentose is 5, hexose is 6, heptose is 7. Their general formula is CnH2-on. Classic examples: fructose from fruit, erythrulose, ribulose, which is key in photosynthesis, and of course glucose, the main energy source for most cells.
Simple, fundamental units like individual Lego bricks. What's next when those bricks start connecting?
Next are the oligosaccharides, which are compound sugars. These form when a small number of monosaccharides, usually 2 to 10, link together.
Okay, short chains.
Short chains, yeah. They yield these individual monosaccharides when you hydrolyze them, break them down with water. If it yields 2 monosaccharides, it's a disaccharide like sucrose, table sugar. If it yields 3, it's a trisaccharide. The general formula for trisaccharides is CnH2ON2, reflecting water loss during formation. Maltose, a disaccharide made of 2 glucose units, is a common example.
And finally, the really big ones, the complex carbs we think of as starches or fiber.
Yes, the third and largest group. Polysaccharides. These are the truly complex sugars. They're polymers that yield more than 10 monosaccharide molecules upon hydrolysis, often thousands.
Thousands?
Yeah, huge molecules. Polysaccharides can be classified further based on what they're made of. If all the monosaccharide units are the same type, it's a homopolysaccharide, like starch or glycogen, both just glucose units linked differently. If they're made of different types of monosaccharides, it's a heteropolysaccharide, like hyaluronic acid in our joints. Their general formula is C6H10O5X, where X is a really big number. Glycogen, how animals store glucose, and cellulose, the main structural part of plant cell walls, are prime examples.
Okay, here's where it gets really interesting. How these different forms, simple monosaccharides, short oligosaccharides, massive polysaccharides, serve such incredibly varied purposes. Monosaccharides for quick energy, oligosaccharides maybe for transport or recognition, and polysaccharides for long-term storage or building sturdy structures. It's a remarkable range of function from such similar basic units. All right, let's move on to proteins. Arguably the most diverse and hardworking biomolecules in any living system. They're the architects, the transporters, the enzymes, the defenders. Pretty much every active process in our bodies involves proteins, doesn't it?
You're absolutely right. They are truly fundamental. The term protein itself is quite telling. It comes from the Greek proteos, meaning preeminent or holding the first place.
Preeminent, okay.
Yeah. This term was first introduced by the Swedish chemist Berzelius in a letter to a Dutch chemist, Mulder, way back in 1838. Mulder then defined them as complex organic nitrogenous substances occurring in the cells of the living beings, and that definition still holds. They're essential intracellular macromolecules, and amazingly, they make up over half the dry weight of most organisms.
Over half the dry weight. Wow, that really underscores their importance. What's their central overarching role in the grand scheme of living matter?
Proteins are found in this central, indispensable position in both the architecture and the functioning of living matter. They're intricately connected with pretty much all phases of the physical and chemical activities that keep a cell alive. Their functions are incredibly diverse. Some provide important structural elements, forming the framework of our bodies. Think keratin in hair, collagen in tendons, even spider silk. Others are vital messengers like hormones, insulin, for example. Many are enzymes, the biological catalysts, speeding up reactions millions of times. They can be oxygen carriers, like hemoglobin, involved in muscle contraction, like actin and myosin. And remarkably, some proteins are intimately related with heredity and genes, playing roles in DNA replication, gene expression.
This incredible versatility, the sheer range of jobs, is what makes them so essential. And when you think about the sheer number of different proteins, just in one organism, let alone all life on Earth, it's mind-boggling.
The numbers are truly astounding. In humans alone, we have about 100,000 different types of proteins, each with a unique, precise 3D structure that dictates its specific function.
100,000.
Yeah. And what's even more remarkable is the species specificity. Like none of the proteins from the bacterium E. coli are exactly similar to any human proteins. It highlights the molecular distinctiveness of different life forms. On a global scale, across maybe 1.5 million species, there could be an incredible 10 to 112 different types of proteins, and maybe 10 different types of nucleic acids. This vast molecular diversity allows for the incredible complexity and specialization of life.
Okay, this raises an important question, though. How can such immense diversity, this entire biological workforce of millions of different tools, arise from just, 20 basic building blocks? How are these countless proteins actually put together?
It's actually a really elegant process. Cells synthesize all their protein components from a relatively small palette of about 20 different amino acids.
That's 20.
Just 20 standard ones, yeah. This synthesis involves multiple dehydration reactions, meaning water molecules are removed as the amino acids link together. The complete structure of a protein is essentially a long chain, or sometimes multiple chains, formed by bonding amino acids together in a very specific sequence.
And what are the key chemical bonds involved in making these long chains and then, crucially, folding them into their functional shapes?
There's a hierarchy of bonds defining protein structure, from the strongest to weaker interactions. The primary bond, forming the very backbone, is the covalent peptide bond. That's ECONH.
The backbone bond.
Exactly. It forms via that dehydration reaction.
So
the carbon atom of a carboxyl COH group of one amino acid bonds with the nitrogen atom of the amino ADH2 group of the next one. This peptide bond is the backbone of the protein chain, dictating the linear sequence of amino acids, which is crucial for its identity.
So that's the strong core linkage, the stable chain. What about the secondary bonds, the ones that add complexity and let these chains fold up?
Right. Secondary bonds are critical for forming those intricate 3D shapes that give proteins their function. One important type is the disulfide bond. There's another type of covalent bond, so it's pretty strong. It's found specifically between amino acid residues in proteins. It forms when the sulfhydryl dehydrogenase groups of two cysteine amino acid residues oxidize, forming a stable covalent bridge called a cysteine molecule. These act like molecular staples, reinforcing the protein's folded structure. Really important for proteins that get secreted or face harsh conditions. Then there's the ubiquitous hydrogen bond. These are weaker, non-covalent interactions. It's where a hydrogen atom, already bonded to an electronegative atom like nitrogen or oxygen, is attracted to another electronegative atom nearby.
Like magnets, almost.
Sort of, yeah, due to slight charges. A common example in proteins is when the carbonyl oxygen of one peptide bond shares electrons slightly with the hydrogen atom of another peptide bond. This helps stabilize structures like alpha helices and beta sheets. We also have ionic bonds, or salt bridges. These are electrostatic attractions between oppositely charged groups on amino acid side chains within the protein. Like charges repel, opposites attract. So a positive lysine side chain might bond with a negative aspartate side chain. Divalent cations, like magnesium, can also form bridges. And finally, nonpolar bonds, usually called hydrophobic interactions. Some amino acids, valine, leucine, alanine, etc., have hydrophobic water-fearing side chains.
They don't like water.
Right. So in water, these nonpolar side chains tend to cluster together in the protein's interior, hiding from the water. They attract water less than water attracts itself via hydrogen bonds, so they get pushed together. This hydrophobic effect is a major driving force in protein folding, burying the nonpolar bits inside.
These various bonds, from the strong covalent backbone to the weaker, flexible hydrogen and ionic bonds, they all work together to orchestrate the protein's final intricate 3D shape, which in turn dictates its precise function. Incredible molecular engineering. And it all starts with the basic units, amino acids.
Indeed. All proteins are macromolecules, big molecules, polymers formed by linking those smaller units, amino acids, the monomers. Amino acids are universally known as the building blocks of proteins. Each amino acid is a nitrogenous compound with the central carbon, the alpha carbon, bonded to four things. A basic amino NH2 group, an acidic carboxyl COH group, a hydrogen atom, and a side chain, denoted as R.
The R group.
The R group is what makes each of the 20 amino acids different. R can be as simple as just another hydrogen atom, like in glycine. The first carbon atom is part of the carboxyl group. The second carbon, the alpha carbon, is connected to the amino group.
So even with those relatively simple components, proteins show immense diversity. How are they classified beyond just their basic makeup to help us make sense of their varied roles?
Right, proteins are similar. Fundamentally, all amino acids link by peptide bonds. But their diversity means we classify them in many ways. By shape, source, composition, solubility, and most functionally by their biological roles. Based on shape, they're broadly split into fibrous and globular.
Okay, tell us about those shapes and what they imply.
Fibrous proteins are, well, fiber-like or long ribbons. They have high axial ratios, meaning much longer than wide, usually 10.
Like ropes?
Like ropes, exactly. Generally animal origin, and notably insoluble in common solvents like water. They mostly play protective and structural roles. Think durable collagen in tendons, keratin in hair. Mechanically, they can stretch and recoil, and they tend to creep or deform slowly under stress. Their long, insoluble nature is perfect for building strong, flexible tissues. In contrast, globular proteins are spherical or ovoid, compact, with low axial ratios, usually less than 10, often just 3 or 4.
Like little balls.
Like little balls, yeah. Generally soluble in water or aqueous solutions, and they diffuse easily, letting them move and interact within cells. Globular proteins are far more complex in their folding than fibrous ones, and this complexity lets them perform a huge variety of dynamic biological functions. Most enzymes, antibodies, transport proteins, they're globular.
That distinction makes perfect sense. Structural ones are long, insoluble, stable. Active ones are compact, soluble, dynamic. What about classifying by source? Does that tell us something about their quality nutritionally?
Yes, from a nutritional view, proteins are often divided into animal and plant proteins. Animal proteins come from animal sources: milk, eggs, fish, meat. These are often called higher quality proteins because they usually contain good amounts of all the essential amino acids, the ones our bodies can't make and we must get from diet. Plant proteins, however, are sometimes called lower quality proteins because they often lack sufficient amounts of one or more of those essential amino acids. They have limiting amino acids, meaning they don't have enough of a particular essential one. The big four, often limited in plants, are threonine, lysine, methionine, and tryptophan. For instance, rice and soybeans can be low in methionine. Ground nuts might lack methionine, threonine, and lysine. Cereal grains are often low in lysine and threonine. Peas and beans might lack methionine and tryptophan.
Okay.
It doesn't mean plant proteins are bad. It just means you need a varied plant-based diet to get all the essentials by combining different sources.
That's a good reminder for anyone thinking about diet and complete protein. Okay, and how about classifying them by their actual biological functions? This seems to really highlight their preeminent nature.
Yeah, this is maybe the most insightful way to categorize them, talking directly about their jobs. First, enzymic proteins, biological catalysts, arguably most critical. Then defense proteins, like antibodies, protecting against invaders. Structural proteins, building and protecting structures. Regulatory proteins, controlling cell activities, like hormones or receptors. Nutrient and storage proteins, providing nutrition, like an egg white, or storing ions, like ferritin for iron. Contractile or motile proteins, enabling movement, like in muscles. There are even toxic proteins, like some snake venoms. And transport or carrier proteins, moving molecules around, like hemoglobin carrying oxygen.
So what does this all mean? This classification truly reveals how proteins are specialized for every imaginable task building, defending, orchestrating life. It highlights their incredible molecular precision.
And just quickly to round out classification, there's also a system based on solubility and composition proposed by physiological societies. It has simple proteins, conjugated proteins, and derived proteins.
Briefly.
Simple proteins are just amino acids linked together, usually globular. Break them down, you only get amino acids. Conjugated proteins are simple proteins linked to a non-protein part, a prosthetic group or cofactor, crucial for function like hemoglobin with its heme group. Break them down, you get amino acids and the prosthetic group. Derived proteins aren't naturally occurring in their final form. They come from altering simple proteins, heat, enzymes, chemicals, or are artificially made polypeptides. It helps distinguish natural versus modified forms.
That helps organize the sheer diversity we're talking about. Okay. Moving from proteins in general to a very special, incredibly dynamic class of proteins enzymes. These are often called life speed demons. And for good reason, right? Without them, life's chemistry would just grind to a halt.
That's an excellent nickname for them, yeah. The term enzyme itself was first introduced by Friedrich Wilhelm Kuhn in 1878 to formally designate these biological catalysts. Before that, they were just known as ferments, linked to things like yeast fermentation. Right. Enzymes are simple or combined proteins acting as highly specific catalysts. They're soluble colloidal molecules made by living cells. All enzymes are globular proteins, so they have that complex, precise 3D structure, which is absolutely critical for how they work.
And the name.
The name enzyme comes from Greek en zyme is yeast, literally in yeast.
Oh.
It reflects one of the earliest reactions observed, making CO2 and alcohol using an enzyme from yeast.
So they were literally discovered through fermentation, which is ancient.
Indeed. Shows how long their effects were seen, even if not understood. Enzymes are fundamentally proteinaceous. Unlike vitamins, animals generally don't synthesize them from scratch. Cells have to make them. Most act inside the cells where they're made endoenzymes or intracellular enzymes running internal processes. But some are made by cells and secreted to act outside exoenzymes or extracellular enzymes. You see this in fungi, bacteria, even some carnivorous plants digesting prey. Our own digestive enzymes in our gut are perfect examples of exoenzymes, breaking down complex food into simple bits for absorption.
So what does this all mean? Enzymes are the essential accelerators. They make life's chemistry possible at speeds that would otherwise be unimaginable millions of times faster. How are these incredible accelerators categorized, given there are so many doing different jobs?
They're systematically classified based on the type of chemical reaction they catalyze. It's a very logical system. 6 main international groups.
Okay, number 1.
1. Oxidoreductases. They catalyze transfers of oxygen, hydrogen atoms, or electrons between substrates. Critical for redox reactions. Examples. Dehydrogenases remove H, oxidases add O, hydrolases. Catalyze hydrolysis breaking compounds by adding water. Crucial for digestion. Examples. Esterases, digestive enzymes. Transferases, catalyze transfer of a specific group methyl, phosphate from one substrate to another. Essential for modifying molecules. Examples. Transaminases, amino groups. Kinases, phosphate groups, often from ATP.
4.
Lyases, catalyze adding a group to a substrate or removing one to form double bonds without hydrolysis or oxidation. Examples. Decarboxylases, remove COH. Aldolases, in sugar metabolism.
5.
Isomerases, catalyze converting a substrate from one isomer to another, rearranging atoms within the molecule. Examples. Fumarase, phosphohexoisomerase.
And 6.
Ligoses, the molecular glue. Catalyze joining 2 molecules by making new bonds, often using ATP energy. Example. Citric acid synthetase in the Krebs cycle.
This systematic organization really helps understand their incredibly precise roles in all those complex metabolic pathways. Each enzyme, a specialized tool for a specific job. And how do we even begin to name them with so many?
Yeah, there are various ways, sometimes a bit confusing but often helpful. Most common. Add the suffix ase to the root word of the substrated axon. Sucrose breaks down sucrose. Lipase hydrolyzes lipids. Fats, right. Some are named based on the reaction they catalyze. Dehydrogenase removes hydrogen. Polymerase aids polymerization, occasionally named from the source papain from papaya. And some older ones keep historical names ending in C, showing their proteins like pepsin and trypsin, digestive enzymes.
That explains familiar names like lactase. Beyond the lab and inside our bodies, what are some biological roles of enzymes in our daily lives that we might encounter without even realizing it?
Well, enzymes have tons of practical applications integrated into daily life, often totally unseen. For instance, in food production, candy making, texture sweetness, bread whitening, breaking starch, essential in wine manufacturing, fermentation clarifying, beyond food, desizing fabrics and textiles, removing starch, even recovering silver from old photofilm. In medicine, specific enzymes for wound healing, removing dead tissue, vital for cheese making, when it coagulates milk. Used to clarify fruit juices, remove cloudiness, and often in supplements or meds to correct digestion, helping break down foods.
What stands out to me is just the sheer breadth of their utility working behind the scenes in so many everyday processes, from food to clothes to healing. What are some of their defining characteristics that make them so powerful and unique?
Enzymes have several outstanding characteristics. First, their particular enzyme action or specificity. They are extraordinarily specific usually.
Lock and key.
Like a lock and key, exactly, with some exceptions. An enzyme works on a particular substrate molecule, often only one optical isomer, mirror image, or maybe a group of structurally similar compounds, but generally highly precise. Second, thermal ability, heat sensitivity. They're delicate. Enzyme action rate usually increases with temperature, up to an optimum, around body temp for us. But beyond that, activity drops fast because heat unravels their 3D structured denaturation, permanently killing activity. Why high fevers are dangerous.
Right.
Third, their colloidal nature. Enzymes are big molecules, diffuse slowly. In water, they form colloidal systems, suspended, not fully dissolved. This helps them interact effectively with substrates in the crowded cell. And finally, their defining trait, catalytic nature. They work catalytically, massively speeding up reactions in tissues without being used up. They just make things happen way faster. Performance is measured by turnover number, how many substrate molecules one enzyme converts to product per unit time when saturated. It can be thousands, even millions per second.
Understanding these characteristics helps grasp both their incredible power and their vulnerabilities, like sensitivity to heat and pH, truly the precision workhorses of the cell. Okay, from the workers' proteins and enzymes, let's shift focus to the blueprints. Nucleic acids. These are the molecules carrying the instructions for building and operating every living thing.
Nucleic acids are absolutely essential biomolecules. Their critical function is storing genetic information in cells. But more than that, they also transfer this info from old cells to new during division and from parents to offspring during reproduction. They are basically the fundamental hereditary originators of living organisms, carrying instructions that make each species unique and ensure life continues. They're macromolecules, big polymers, found in most living cells, either bound to proteins and chromosomes or free. Like proteins, they have high molecular weight and are made of repeating smaller units called mononucleotides.
And what are the two main types of nucleic acids carrying out these vital roles? We hear about them all the time.
Right, two primary types, working together but distinct. Deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Both are universally present in all plants and animals, the basis of their genetic systems. Worth noting, viruses usually have either DNA or RNA as genetic material, generally not both.
Let's start with DNA, the master blueprint. What makes it so crucial as life's central information store?
DNA is the undisputed genetic material for all cellular life. It's the master instruction manual differentiating each species. This incredible genetic code is meticulously passed on through reproduction, from adults to offspring, ensuring traits are inherited. In eukaryotic cells, with a nucleus, DNA is mostly in the chromosomes, tightly packaged structures in the nucleus. A small, vital amount is also in mitochondria, the cell's powerhouses, which have their own DNA.
Right, mitochondrial DNA.
Exactly. DNA is indeed tightly packaged, folded, and coiled immensely to fit inside the tiny nucleus. It has that characteristic coiled structure. Its functional units are segments called genes, each holding specific instructions for building proteins or regulating things. The iconic shape is the double helix, two polynucleotide strands coiled around each other, like a twisted ladder. In sexual reproduction, offspring get half their DNA from the male parent, half from the female, create variation. While usually double-stranded in some viruses and rare organisms, DNA can be single-stranded.
So DNA is the long-term, tightly packed master instruction manual, the archive. What about RNA, often called the versatile messenger and worker? How does it complement DNA?
RNA, ribonucleic acid, performs several crucial diverse jobs, working directly from DNA's instructions. These include gene expression, using genetic info to make proteins, and gene regulation, RNA molecules switching genes on-off. It's involved in coding, decoding, regulation, expression. That's lots of roles. But its main, most famous role is facilitating protein formation, acting as the intermediary between DNA and the protein-making machinery, ribosomes. Unlike DNA, RNA is typically made of single long strands of nucleic acids. Each RNA nucleotide has a base, adenine, cytosine, guanine, or uracil, ribose sugar, and a phosphate group.
The worker bee.
Yeah, you can think of RNA as the diligent worker sending the DNA message out to the rest of the cell, carrying instructions to the ribosomes. While usually single-stranded, interestingly, transfer RNA, tRNA, folds back on itself in specific ways, forming 4 regions of double-strandedness, giving it a crucial L shape for its job in protein synthesis.
So while they both carry genetic info, there are key differences in the molecular structure, even though they share similar parts. What are those crucial distinctions?
Absolutely. Subtle differences lead to vastly different roles. Let's compare. Both DNA and RNA have phosphoric acid, forming the backbone. The key difference is the pentose sugar. RNA has D-ribose with an OH on the 2-met carbon. DNA has D2-deoxyribose, lacking that OH on the 2-met carbon, hence deoxy. Right. This small difference makes DNA more stable, better for long-term storage. RNA's extra OH makes it more reactive, transient. For the purines, double-ring bases, both have adenine and guanine, but for the pyrimidines, single-ring bases, there's a vital difference. RNA has uracil and cytosine. DNA has thymine and cytosine.
T in DNA, U in RNA.
Exactly. So in DNA, A pairs with T. In RNA, A pairs with U. This T for U swap in DNA also adds to its stability and helps with repair mechanisms.
This subtle shift in just a few components makes a world of difference in their roles. Stable blueprint versus versatile messenger. Elegant structure-function relationship. And what about nucleosides? How do they fit in?
Nucleosides are the base units before the phosphate gets added. They're purine or pyrimidine base linked to a sugar, ribose or deoxyribose.
Base plus sugar?
Base plus sugar, yeah. If it has ribose, it's a ribonucleoside. Deoxyribose makes it a deoxyribonucleoside. They form when a base reacts with a sugar, losing a water molecule. Example, adenine plus ribose gives adenosine, ADEC. Adenine plus deoxyribose gives deoxydenosine, DA. The purines are adenine, guanine. Pyrimidines are cytosine, thymine, DNA, uracil, RNA.
And then adding a phosphate to a nucleoside gets us to nucleotides, the actual monomers of nucleic acids.
Exactly. Nucleotides are the complete monomers. They're a nucleoside plus a phosphate group or groups attached to the sugar, usually at the C5 position, but sometimes C2 but or C3. But they exist free in the cell, like ATP and energy carrier, or linked together as subunits in nucleic acid chains. Crucially, the sequence of an RNA chain is directly dictated by the complementary sequence in the cell's DNA. That's the central dogma of molecular biology in action.
If we connect this to the bigger picture, these nucleotides are the individual letters spelling out all of life's instructions. Whether stored in the DNA archive or carried out by RNA, they're the fundamental units of biological information. A remarkable system. Okay, let's transition to another vital class of biomolecules. Vitamins. Small, but incredibly mighty. Absolutely indispensable for our health, even though we only need tiny amounts.
Vitamins are indeed fascinating. They're indispensable for normal development, growth, and maintenance of animal organisms, including us. They're found in plants and animals, but typically only in small, trace amounts in food. What's crucial is that most animal cells generally don't make them. We have to get them from outside, mainly through diet.
Right, gotta eat them.
Mostly, yeah. Though there are exceptions. Some vitamins get synthesized in our intestines by our gut bacteria. Overall, they're required for good health and all sorts of life functions. A vitamin is specifically defined as an organic compound, essential as a nutrient in small amounts. Interestingly, our bodies can synthesize a couple. Vitamin D, when skin gets UV light, and vitamin A from its precursor, carotene, found in orange-yellow veggies.
So we make a couple, but mostly rely on diet. That emphasizes needing a balanced diet. What are some key players here, and what critical jobs do they do? Maybe listeners can think about their own diet as we go through these.
Definitely. Let's look at some common ones.
Found in orange yellow fruits veg, liver, dairy, oatmeal, crucial for skin hair maintenance, eyesight, especially low light, bone tooth growth.
The sunshine vitamin, also in fortified milk, fatty fish, eggs, butter. Vital for strong teeth bones, calcium absorption, helps heart action, nerve system. Vitamin E. For multigrain cereals, wheat germ, vegetable oils, nuts. Primarily a powerful antioxidant protecting cells and tissues from damage, maintains fatty acids.
Fruits, dairy, especially green leafy veg, spinach, kale, main job. Essential for blood clotting functions. Vitamin B6 in fish, poultry, bananas, beans, whole grains, lean meats. Builds tissues, key coenzyme in protein, carb, fat metabolism.
Mostly animal products, meat, seafood, milk, crucial for nerve system function, carb fat metabolism, DNA synthesis, red blood cell formation, a concern for strict vegans,
Right.
and vitamin C. Abundant in citrus fruits, many veggies. Essential for structure of bones cartilage, iron absorption, boosts immunity, improves heart functions.
That's a great overview of the individual stars.
Yeah.
How are vitamins generally classified into broader categories, that fat-soluble, water-soluble thing?
Exactly. Vitamins are fundamentally identified in two main classes based on solubility, fat-soluble and water-soluble.
Yeah.
This distinction is crucial. It dictates how our bodies absorb, transport, store, and excrete them.
Okay, let's start with fat-soluble. I imagine these get stored in our fat.
Precisely. The fat-soluble vitamins are A, D, E, and K. They're oily substances, not easily soluble in water. Their biochemical functions are complex, but they share traits. They only contain C, H, and O, and share close chemical similarities, often isoprenoid derivatives. Key point.
Yeah.
They can be stored in the body, mainly in fat tissues and the liver.
So you can build up reserves?
You can't. The liver can store months' worth of vitamin A. All vitamins are absorbed in the small intestine, but fat-soluble ones specifically in the duodenum, often with dietary fats. A is mostly liver, K and E separately in liver, D mostly in fat and muscle.
So those are the ones we can stockpile a bit. And the water-soluble ones, I assume those are the opposite, need more frequent intake.
Exactly. Water-soluble includes the entire B-complex group, B1, B2, B3, B5, B6, B7, B9, B12, plus vitamin C, also often included. Inositol, choline, papain A, lipoic acid, bioflavonoids. These often act as catalytic factors, vital links in biochemical reaction chains, frequently as coenzymes. B-complex vitamins, being water-soluble, are excretable, not stored significantly. Excess leaves via urine. So small daily intake is needed for normal function.
Use them or lose them.
Pretty much. They're absorbed with water, mixed right into the bloodstream. Because they aren't stored well, consistent daily intake is important to avoid deficiencies. Vitamin C, mainly stored in the adrenal cortex, can show deficiency symptoms, like scurvy, within just a week or two if intake stops, showing its rapid turnover.
This distinction dictates how our bodies handle them: absorption, use, storage. Explains daily allowances for water-soluble, maybe more caution with high doses of fat-soluble. What are the overarching functions of vitamins generally, tying all these specific roles together?
Okay, summarizing the big picture. Fat-soluble, ADEK. Vital for vision, immune function, skin, A. Calcium absorption, strong bones teeth, D. Antioxidant cell protection, E. Blood clotting, bone metabolism, K. Water-soluble, B-complex, C. Indispensable for energy metabolism, food to energy, countless enzyme functions, B-complex. Red blood cell formation, nervous system health, B-complex. DNA synthesis, brain function, B-complex. Boosts immunity, antioxidant, C. Collagen synthesis for healthy skin, vessels, wound healing, C.
Right.
Across the board, all vitamins act as coenzymes or cofactors, helping enzymes work efficiently. They collectively support growth, repair, maintenance of body tissues. Crucially, they prevent specific deficiency diseases, scurvy, C. Anemia B, rickets D. Ultimately, sufficient amounts promote overall health and well-being.
It's clear they're tiny, but their impact is absolutely vital. Essential co-pilots for our internal biochemistry. Okay, our final deep dive brings us to metabolism. The grand orchestrator of all these biochemical processes we've talked about. Atoms, bonds, water, carbs, proteins, enzymes, nucleic acids. Metabolism is literally the engine of life itself.
Metabolism is indeed the ultimate biochemical symphony. It refers to the entire group of biochemical reactions organisms carry out, serving two main purposes. First, synthesizing all necessary cellular substances, building things up. And second, utilizing energy from their environment, powering everything.
Building and powering.
Right. It can be concisely defined as the sum of all enzymatic reactions in an organism or cell. At its heart, it's the process where the living body converts nutrients into usable energy. During this complex biochemical reaction, calories from food combine with oxygen, like Lavoisier, found with respiration to produce the energy needed for every bodily function. And we need this energy continuously for everything. Breathing, adjusting hormones, circulating blood, growing, maintaining and repairing cells. Without metabolism, life stops.
So it's not just an engine, it's a self-sustaining, self-repairing, self-regulating engine. What are the four main functions that metabolism enables in all cells? The core purposes.
In every living cell, metabolism enables these vital functions. Four main ones.
Converting nutrient molecules into precursors for cellular macromolecules, breaking down raw materials into building blocks.
Gaining chemical energy from degrading energy-rich nutrients or from solar energy, harvesting power.
Okay.
3. Synthesizing and breaking down biomolecules needed for specific cell functions. Constant recycling and creation of specialized molecules.
Building these precursors into complex macromolecules, proteins, polysaccharides, lipids, nucleic acids, etc. It's a continuous cycle of transforming and constructing.
Okay, here's where it gets really interesting. It's this continuous dynamic cycle of building up and breaking down, often happening at the same time in the same cell. How is metabolism typically classified to help us understand this interplay?
Metabolism is elegantly divided into two major interconnected classes: catabolism and anabolism.
Catabolism, breaking down.
Exactly. Catabolism is the breaking down phase. Its main function is releasing chemical energy informed cells can use for chemical and mechanical work. It breaks down complex organic compounds into simpler ones. Think of it as the cell's demolition crew. Catabolism provides energy through key pathways like glycolysis, breaking glucose, the Krebs cycle, central energy hub, and the electron transport chain where most ATP is made. This energy is captured mainly as ATP.
So catabolism, breaking down, releasing energy,
right.
And anabolism must be the building up side.
Exactly. Anabolism is the building up phase. Its main function is using the energy from catabolism for biosynthesis, creating cell elements. Think of it as the cell's construction crew.
Got it.
Photosynthesis in plants is a macro-level example. Anabolic pathways involve several enzyme steps, resulting in synthesizing complex macromolecules from the simpler intermediates made by catabolism. So catabolism provides energy and raw materials. Anabolism uses them to build and repair. These two are constantly in dynamic balance.
So think of it as the demolition crew and the construction crew of the cell working in perfect sync, constantly recycling and rebuilding. And all this breaking down, building up requires energy. Which brings us to a molecule we've mentioned a few times, ATP.
Indeed. ATP, adenosine triphosphate, is absolutely central. In us, or any living thing, energy is stored and transferred primarily as ATP. It's the universal energy currency of the cell. Its direct use is universal and constant. Anytime muscles contract, stuff moves across membranes, nerve signals fire, an enzyme is made. Our body uses energy straight from ATP hydrolysis. That's where ATP loses a phosphate group, becoming ADP, adenosine diphosphate. This hydrolysis releases a significant energy burst, about 7.3 kilocalories per mole, immediately available for cellular work.
So what does this all mean? This single molecule, ATP, powers almost every single process we've talked about today, from the smallest bond formation to the grand symphony of metabolism itself. It ensures cells can build, move, think, survive. Truly the universal energy currency of life, linking everything. Hashtag, tag, outro. Wow, what an incredible journey today. We went from the foundational atoms and chemical bonds holding everything together through water's essential role as life solvent. Explore the major biomolecules, carbs, proteins, enzymes, nucleic acids, each playing these distinct interconnected roles.
Of quite a tour.
Yeah. And then we tied it all together with metabolism, that dynamic, relentless engine powering everything from a single cell to us. You hopefully now have a solid, detailed foundation in the chemical processes underpinning all living organisms. We really aim to make this deep dive a shortcut to being truly well-informed, packed with surprising facts and crucial insights into life's very nature.
And, this raises an important question, something to maybe think about beyond today. Considering this intricate dance of chemistry sustaining life at every level, from tiny molecules to whole ecosystems, how might a deeper, more profound understanding of these biochemical fundamentals empower us? How could it help us approach the big challenges in health, environment, technology, maybe in an entirely new, even revolutionary ways?
That's a great question.
It really highlights the constant learning and how all knowledge is interconnected, urging us to keep looking deeper.
A powerful thought to leave you with. The more we understand life's chemistry, the more we can potentially shape our future. Thank you so much for joining us on this deep dive. This deep dive was presented by Knowledge Flow. We invite you to continue your learning journey with us. Until next time.